All right, everyone, thanks for coming to the December installment of the Delaware Valley Mass Spec Discussion Group meeting. Before we get started, what I wanted to do was just give a shout to the sponsors who are responsible. And if I could also reiterate that starting in 2022, we're hoping, if everything cooperates with variants and vaccines and everything, that we can get back to an in-person meeting, certainly for the vendor night, which would be in the late spring, early summer. So what I want to do is thank our sponsors and reiterate that we would really like them to get back to going with their normal dues and contributions that allow that to happen. So everyone who's listed here: Mobile Ion, Agilent, Bruker, Gentech, IonSense, Geol, Keystone, LECO, New Objective, NEMS, Phoenix S&T, Restec, SciEx, Shimadzu, Adaptis, Thermo, Trajan, and Waters. Thank you all for supporting us in the past, and let's get back to it. And I also want to try and encourage everyone to keep an eye on LinkedIn, on Instagram, and on YouTube for all of the Delaware Valley Mass Spec Discussion Group and all of the things that we're working on. So I'm going to stop sharing now, and I'll hand it over to Abraham, and he can get ready while I pull up his introduction. It's a real pleasure to have Abraham Badu-Tawia here with us today. He got his PhD in 2012 from Purdue University under Graham Cooks. After that, for a couple of years, he was a postdoctoral fellow under George Whitesides, then joined the faculty at Ohio State University in the Department of Chemistry, where he is now an associate professor and a recipient of the 2021 Ohio State Early Career Innovator of the Year Award, the 2020 Sloan Fellowship Award, the 2019 NIH Mira for New Investigators Award, 2018 ACS Division of Analytical Chemistry Arthur Findace Award, 2017 Eli Lilly Young Investor Award in Analytical Chemistry, and then the 2017 ASMS Research Award. His current research is focused on the development of new mass spec techniques for disease detection and the studies of novel ion chemistry and charged microdroplets. And I'm very excited to hear about this particular talk because I think it's very important. And I'm really looking forward to hearing more about science in reverse. So please take it away, Abraham. Thank you.
Thank you so much, Joe, for the kind introduction. Thank you, Hash, for also inviting me to give a talk here. I will say that I'm also the organizer for the Ohio State Mass Spec Discussion Group, and we haven't had a lot of funding yet. So I'll talk to you later to see how you're able to pull all this funding together to support your group. Very nice. Well, so today I thought I would share with you the new developments in our lab. And as I've told Joe before, most of these are not published yet, but hopefully we can have a good discussion. And in fact, you can actually stop me and I'll be happy to go a little more details or answer questions as we go. So science in reverse, and I'll explain what I mean in a moment, but since I got this opportunity, I thought, I will first introduce my lab in a broader sense, what we do, and then I'll pick two of the projects going on to discuss today. And then as part of the science in the reverse, I'll talk about a new invention we made in the lab, which we are calling panoptic mass spectrometry. The idea of the panoptic is to develop a universal ion source that is applicable to almost any analyte type of interest, being it small or large, polar or non-polar, complex or pure samples. And I'll also show you the same source is able to give us 4-quadrant mass spectrometry. As you know, today we have only 2 modes of mass spec. Think about how we can, if you can have 4 modes, of the mass spectrometry, how that can enable improvements, in chemical analysis. And then I will switch gears and talk about immunoassay experiment. And in fact, I will explain later about it. The science in the reverse really is targeting the underserved population who have limited resources. And so if I can have a single device that can do a whole bunch of other things, then I can have it in a resource limited settings, and that will serve millions of people. Likewise, if I have a tool like immunoassay that is sensitive, I can use it, to diagnose diseases, and I'll use malaria as an example today for asymptomatic patients, those people that are walking around as if they are okay, but they are carrying a parasite. I think the COVID pandemic now causes us to understand or appreciate, infections in different levels. And most importantly, and I try to make an application in terms of teaching. So as a way of introduction, this are the 4 main areas of research in my lab. As Joe mentioned, my PhD focused heavily on droplet chemistry. We are still working on this. Unfortunately, I'm going to skip this today, and I'll give just one slide summary. And then we also have these macrosampling platforms. And because we want to go out to the field, to the underserved groups, we are careful in preserving the sample that we collect in the native state, even without any cold storage. So this is also a major area in my lab, which I will also mention briefly, just maybe one slide. And then I will jump into the project number 2 and 3 in more details. And so the droplet chemistry, I would say it is the most productive project in my lab, where we are using charged micro droplets as reaction vessels for chemical reaction. And we are using electrospray, and we spray these droplets across a certain distance, maybe 20 centimeters. And across this distance, these droplets shrink in size, and they concentrate the reagents into small space. That allows reaction to go faster. Also, as the solvent evaporates, you have some reagents sitting at the surface of the droplets. Those are solvent limited species that allows reaction also to go faster. So we've done all this fundamental work, but right now what we are doing, I mentioned this earlier, that we are coupling the droplet chemistry between an LC, and a mass spectrometry, whereas you separate your species as they come out, you do the droplet reaction in real time on a microseconds time scale. You change the identity of the reagents or the analytes and make them more detectable in a mass spectrometer. And this is working well. An example is sugars. Since this is a mass spec group, I know this is quite difficult to actually get sugars to ionize by themselves. This is a 7 mixture monosaccharides and disaccharides. Some of them you can't even see. But when you do the reaction, as they are coming out from the LC, all of them shows up really sensitively. And we are getting down to picomolar sensitivities with the reaction. The other project I'm going to skip today, just so that you can have a feel for what we are doing in the lab, is this Macrosampling Platform. And I will talk more about malaria today, but the gold standard for diagnosing malaria is microscopy, thin film or thick film microscopy. But unfortunately, you cannot use dry blood for microscopy. And the reason is most dry blood are collected in paper. And if you use hydrophilic paper to absorb the blood, and this is what you see. Within 10 minutes of collecting the blood sample, you see the red blood cells, almost all of them, except with the exception of a few of them, they all lies up. In fact, within 1 hour, you can see them kind of expanding in size. That is water diffusing into the red cells. And then 2 hours, you cannot see any red cells. And the microscope literally is counting the infected red cells. So if you can't see them, there's no chance, to diagnose malaria using samples collected from the field that are stored dry. In fact, this has been a major area of investigation for almost 100 years now. It's not been solved yet. What we proposed to do was to change the paper from hydrophilic to hydrophobic, so that when you put the blood on the hydrophobic paper, it builds up and it forms a sphere. Yeah, so we are calling it blood spheroid. And if you allow this blood spheroid to dry, it dry as a dry blood spheroid, still keeping the shape. Now, it turns out that on the surface of the sphere or the spheroid, within some few minutes, maybe an hour and a half, as the water evaporates, okay, the red cells come together, but you can see that they are bridging themselves. This mechanism has been proposed a while back. I think this is the first time that we are seeing the red cells really bridging and then forming the self-assembled, red cells as the solvent evaporates. So within 1 hour, the solvent evaporates, and you're seeing this on the surface, red cell self-assembly. Well, if you leave the blood in ambient air for another 20 minutes, what you see is that all the surface red cells, now they are melting, they are lysing up, and surprisingly, when they lyse, they form this thin surface layer, and when you crack it open, what you will see is intact red cells in the interior of the spheroid. So although all the surface red cells are gone, the interior red cells are still intact. And so what you can do is actually, put this in water, reconstitute it, and get the red cells back again. And now count the infected cells with your microscope. And so we are pushing this also heavily. We are developing sort of machine learning tools that will allow the counting process on the blood, the dry blood, to be more effective. And I have to skip this, unfortunately, today. But I'm going to focus on is the two projects that I discussed earlier. So what do I mean by science in reverse? And I'm relating this science in reverse to writing in reverse. Writing in reverse, I think, means writing with a recipient in your mind. And so doing research with somebody or someone specific in mind, you are doing it in reverse. And the reason why that is important is because when you have that person in mind, those group in mind, you are able to design the experiment to fit their need much more effectively than just doing it the way we do it typically. Like I'm a mass spec person and I design my experiment based on my expertise, not really thinking about who will use it next. In academia, this is very common that we don't do it in the reverse. Of course, I'm not the first person to think about this. The WHO, in terms of the underserved group, have defined some criteria that we must meet, when you want to design an experiment for the underserved. And the big thing for them is that they don't want you to use, an equipment. And so under this assured criteria, the best way to meet it is to do a rapid diagnostic test, where you take a plastic or a piece of paper, you put antibodies in lines, and then you put your sample there, and as the sample works through the paper or the plastic, a line will develop or will show up that will tell you, yeah, you've captured some biomarker is positive or negative. Okay, now that's great, but this approach, although it's a reverse, I want to say that it's working in a normal mode. Normal mode, meaning that when you go to the underserved population, yeah, developing countries, Southeast Asia, we know that those people are poor. And so the method we are developing for them, we say it has to be cheap. And guess what? The cheap method or the low cost method typically comes out with low value. And anytime we are thinking, in the reverse or in a normal mode, we think that a high cost method usually brings high value. Okay. And so what I really mean by the reverse is, yeah, we are thinking from the person's perspective, but how can I have a low cost method with a high value instead of a high cost with a high value? If I can achieve this, a low cost method with a high value, then I'm working in the reverse mode, also in the reverse, scientifically. And the reason why this low cost, high value is important is illustrated in this data. But this is the current data that was brought to my attention by a medical doctor. He told me that today, young adults, okay, less than 50 years old, we are seeing incident rates in colorectal cancer. We focus for so long on adults, older adults, okay, and we work very hard on reducing the mortality rates. But in the young adult, people that perceive themselves to be not at risk of cancer, how can we get them, to test themselves or to go for screening as early as possible? Because if we do that, then we have the chance for treating them, more effectively. And today, and the way that we reach them is through this low cost, low value approach, which is a fecal immunochemical test. This test, as it stands today, is only 30% sensitive towards early diagnosis. That means that if you use it, you are missing 70% of the people that could be at risk. And so what can we do? Can we have a low cost? People that, again, perceive themselves to be low risk, okay, they won't do it if the cost is high. But while they are keeping the cost low, can we have a better sensitivity than 30%? So that's a kind of a conundrum, it's a challenge. And we've kind of dedicated ourselves for solving this problem. And the technique that I'll discuss today is applicable, of course, in the US. It's also applicable, in the developing countries. And that's where I started from. So the way that I think this can be solved or to achieve low cost, high value, in my opinion, is to integrate multiple devices into one platform, so that it's a single platform that is doing a lot of things at the same time, so that I don't have to go through multiple chemical detection platforms to achieve the sensitivity that I need. As we add more and more and more platforms, more and more methods, we increase the cost. So at least if I can have this device, I cut down the cost and achieve it. And so the first thing that comes to my mind is using the portable instrument. Okay. And of course, there are obvious limitations for portable instrument. For example, the mass range. You cannot use a portable instrument to detect proteins. But can we? Maybe, yes. Most portable instruments, we cannot couple a liquid chromatography to them. So what can we do? Taking a portable instrument, how can we increase its value and make it do more than it typically does? If I can achieve that goal, then yeah, I'm using a portable, simple instrument that now have a much more value than traditionally is. So let me illustrate what I mean. And so I'll come back, to the malaria, but now I want to go through to kind of describe our effort to empower the portable mass spectrometer to do more than it typically does. If we can achieve that, then we can take it to the field and then begin to work with real people. Okay, so collectively, we can group chemical species that we want to detect, into different groups: solids, liquids, polar, non-polar, gas, solution, all of this. And I'm sure we all know that in terms of mass spectrometry, if I have a polar compound, it works really well for, if I use ESI. If I have non-polar, ESI fails miserably. So the recommendation is use APCI. Okay. If I have a protein, ESI also works really well, but the information that I'm seeking actually determines a specific experiment I have to do with the ESI. Am I looking for a native, information which is conformationally important? Then I have to do, native mass spectrometry. Do I want the sequence information? That's a different kind of experiment, although the same process. If I have a complex mixture and I don't have a liquid chromatography, can I still analyze this complex mixture without any sound preparation? Yes, that will be ambient mass spectrometry. So all of these are out there. Most of them are not commercially available. You can buy them one by one and do whatever you want, depending on sample that you have. Can I have one source that collectively can do all of this without switching between them? And if I gave this talk recently, I was being considered for promotion to a full professor. And so I wanted to emphasize that although this slide seems simplistic, most of the techniques I mentioned here represented a breakthrough, in the field. ESI was a science paper. DESI, ambient mass spectrometry, was a science paper. Native and in proteomics, all of them are high quality experiments. And I argue that it includes even Horning's experiments, who used APCI for the first time to couple to liquid chromatography, which was not possible with ESI. I actually think that ESI at that time didn't exist. So that was a breakthrough. Now imagine if you can have one source that can do all of this together. I thought that would be an important process. So how does this look like? The thing that I want to use to analyze all kinds of analysis comes down to this device, which we are calling panoptic source. I want to say that if we are developing a prototype right now, which we can attach to any portable, any instrument for analysis. This device has actually two main modes, a single mode and a hybrid mode. The single mode can be operated in 3 independent types. And that's illustrated in the 3 emitters here. The first emitter is sort of a traditional ESI or nano ESI, where your solution is contained in a glass with a sharp tip. And then you come in with an electrode, maybe a silver electrode. You apply voltage, maybe 1 or 2 kilovolts. Then you charge your solution, and then you spray it. That is a contact ESI. We've shown that in fact you don't need the contact mode of analysis. You can do a non-contact nano ESI, and I'll show you on the next slide that the non-contact actually has some benefits. For example, it reduces heating, so that if you really want a native, state mass spectrometry, you don't want the high voltage in contact, with the solution of the analyte. And so then Non-contact is another way, another single mode analysis. And then if you had a solid sample or even a volatile species, okay, and you wanted to do APCI, you can apply 4 kilovolts to this electrode number 3, and a small plasma will be generated here, and you can do an APCI from this vapor or from the solid. I have movies, I didn't add it to this slide. I have movies showing that in the presence of the voltage, if you have a powder without any physical contact, you could actually release the particles of the powder into air and now ionize the particles using the plasma. I think I have one data for this for you. And so all of this can be operated independently. That's 3 modes, 3 single modes. And then you can combine them also together to have the hybrid modes. For example, on the first slide, I show you that we can do droplet chemistry. So I can fuse 2 droplets together, one reagent here, another reagent there, and that will react in real time to change the properties of your analyte. That will give you the dual nano ESI mode when you combine 1 and 2. You can also achieve separation. For example, on a portable instrument, you can achieve electrophoric separation when you use 2 and 3. And I'll show you an example as I come along. And then you can also actually do some gas phase chemistry. I think I have one example here to show. Also, you combine 2 and 3, but in a different way. Okay, so the same source is doing all of these for proteins, for non-polar, for polar, complex mixture, and all of that. So let's see. Okay, so I'm showing you data here for solid powder of these 2 compounds. Here's vitamin D. They are placed in container C. Okay, they are fine powder. If you apply a voltage 4 kilovolts into this, you release the powder and you ionize these particles. It works very well. There's no physical contact, and yet it works. If you have a protein, so here is a protein in water. So this is a pure solution of protein. I want a native mass spectrometer from this. Okay, I put it in this number 2. There's no contact, and I apply maybe 2 kilovolts or 1.5 kilovolts. I can spray it, and I get my native data. The charge state tells me that the protein is in a native state. Now I want to denature the protein that I have here. In fact, we've measured the volume of this protein. You can load about few picoliters of protein, on this tip. So if I wanted to, denature it in solution, it will be difficult for me to manipulate that small volume. But, in this source, all I do is I have an acid vapor that comes here, and it gets collected into the solution of the analyte. And if you leave it for 10 seconds of seeding the vapor into the solution, the charge state clearly is changing, meaning that the protein is also changing the conformation. The more charges you have, the more unfolding the protein has done. Now you can do proteomics on this pretty well. If you leave it for 45 seconds, the protein has fully unfolded, more charges can be added. Now this is easy because the protein is in water. Now if you take a complex mixture in the same protein in PBS buffer, if you do electrospray of this PBS buffer, I know many of you might have experienced this. It will not work unless you do some other things with it. Okay, but we take the same solution, we put it here, but instead of analyzing right away, we first apply a negative voltage, okay, to electrode number 2. So negative high voltage, okay, 5 kilovolts, negative 5 kilovolts to this electrode. And what happens is that all the cations, in the solution of the analyte see this negative voltage and they are moving towards it. And so the very tip, okay, of the capillary here is depleted of your cations. Now we reverse the voltage to positive. And then we are using 2 kilovolts positive. Then we turn on the mass spec to scan at that time. The big proteins who did not respond very well, to the higher voltage are still at the tip. Now that we are spraying and collecting the ions, is the protein that come out first. You see it, they come out. Then as you keep spraying, surprisingly, we are beginning to see different conformations of proteins coming out. And then as you keep spraying, all the bigger proteins go out. Now the native ones come in. And I'm willing to discuss, with you why we are seeing different charge states. Clearly, we are seeing 2 different conformations. Does this exist already in solution, or something actually happened? And we believe that something might have happened, during the analysis. For example, I told you that we apply negative 5 kilovolts. Now, if you apply negative or 5 kilovolts, in contact with your solution, the tip of your capillary actually bends. There's so much heat that generates that the tip actually can break if you increase the voltage. So this is the traditional contact mode. But if you pull the electrode out from solution and you do a non-contact, the heating is reduced, but still you can see that the capillary is still not as clear as you want. Some heating going on. And so that heating that occurs at the 10 seconds of trying to deplete the cations can actually cause unfolding in solution. Okay, but now look at what happens. When you have this second electrode in close proximity, to the tip, okay, that means that the 4 kilovolts or the 5 kilovolt is being applied to the 2 of them, and we are generating plasma. When that happens, you have a much cleaner or much more stabilized tips. And that's why we are saying that you need to couple the non-contact ESI number 2 to APCI number 3. It's under that condition that you can actually get this data. If you just do a non-contact in the absence of number 3, you still can't get in data because the tip are already destroyed. And so this hybrid mode is what now allows us effectively to, number 1, sort of deplete the ions from the tip while also stabilizing the tip by reducing the heating. And again, if we have time, I'm willing to show you some movies that actually shows that there's a lot of heat when this happens, but the corona discharge actually acts as a cooling effect that stabilizes the tip. If you have questions now, you could please stop me and ask.
Yeah, this is Harsh. I have a question regarding this corona discharge.
Yes.
So you mentioned that you're seeing a conformational change due to removal of charges or reduce heating effects.
Yes.
But the discharge itself, do you think will cause charge reduction, like proton transfer kind of chemistry to reduce the charge? Instead of the conformation,
yes,
the overall charge density of the ions, would you expect charge transfer chemistry?
That's a very good question. In fact, I know people like Smith in Wisconsin has exposed protein ions to corona discharge, and he sees a charge reduction. And that's possible, but I doubt, and that's what's happening here. I doubt it because if that's the case, for example, when we start spraying here, in fact, we are using like 2 kilovolts, and the breakdown voltage for air is actually 4 kilovolts. So when we begin to scan the mass spec and we are spraying, there's no discharge. Okay, the discharge only happens during the depletion regime. And so at the time that we are scanning the mass spec, we should see the real proteins themselves, which suggests that these ions we are seeing might be coming from solution, but they might not be there originally, because if you took the same solution, in fact, this is the same one, and you spray it, this is what you get, And so by using the high voltage, perhaps it's what has changed the conformation in solution, even within the 10 seconds before we begin to collect the mass spec. Yes.
Great. Yeah, that was kind of the first demonstration of a charge reduction.
Right, yes,
Using
right.
the
Yes,
right.
and I would say that, in fact, we tried it. We've tried, so we tried the corona discharge. So right now, in fact, we have so many homemade sources that are coupled to corona discharge. And I've tried to sort of treat the proteins, with the corona, and it doesn't work that well. It doesn't work that well. I met Evan Williams some time ago at a conference, and he also was kind of doing the same thing. It didn't work for him either. I think that Lloyd Smith might have evaporated the droplets before he exposed them to the corona. Yes. Right. And so here's a data that I think blows my mind, actually, how this thing works. So if you take a small molecule, okay, this is 15 different compounds mixed together. Traditionally, you have to do LC or something to separate them and then ionize them. Otherwise, if you do direct analysis like we've done here in spectrum A, we can only see four out of the 14 of these. There's a significant ion separation. But now if you begin to do the electrophoretic separation, we first apply -4 kilovolts to the same emitter number 2. We wait for 10 seconds. Then we apply +4 kilovolts. Okay, so this is different, slightly different from the previous one. The previous one was 2 kilovolts analysis. So the plasma is turned off. But this one, we're intentionally applying 4 kilovolts. So there's a plasma there. That's because in this mixture, there's polar and there's non-polar. So if you don't have plasma, all you can see is only the polar compounds. But in the presence of the plasma, then you can ionize the non-polar species as well. And so the first step is to separate these species in space. So you apply the negative, and these ions that are sitting here, they kind of see it and they spread themselves in space. Then you pulse it with the positive kilovolts. Then they come out one by one while they're experiencing the plasma. And all of a sudden, 13 out of these 14 can be seen. And when you average over the entire spectrum, it's what I'm showing you here. The only compound we couldn't see is a sugar, sucrose, 13. Very difficult to see the sugars. And we had to do some reactions with it. I put chloroform here, and the chloroform actually reacts with the plasma to generate chloride anions. Now the chloride attaches to the sucrose, and then when you flip to the negative mode, then you can actually see the sucrose also. So, from the same source, you can analyze all this complex mixture without a dedicated, separation. The source itself is doing the separation for you, while you're also analyzing the polar and the non-polar. And so if I can have this functional device and have one portable mass spec, in some developing country, I can do a lot of work with this source, with the reasonable, resources there. So that sort of brings me to the one fourth, of the talk. Now I'm kind of going to show you And the other aspect, maybe the other novel aspect of the same source, we learned that the same source actually allows us to do a 4-quadrant mass spectrometry instead of 2-mode mass spectrometry. And what I mean is that traditionally, we know that if your source is in a positive mode, we ask you to scan your mass spec in the positive mode. If your source is in a negative mode, we say scan your mass spec, in the negative mode. So all we have is just 2 modes. It turns out that in the presence of the plasma, although your source might be in a positive mode, you are actually generating negative ions. So you can scan your mass spec in the mismatch polarity, positive and negative. You can also, whilst the source is in the negative, you can still scan your mass spec in the positive mode and get data. And so to illustrate this, that's what I mean. Today we have positive, positive, negative, negative, 2 modes. We assume that if your source is positive, you can only generate positive ions. But what we have learned through this experiment is that even though your source might be in positive, you can still generate negative and positive ions. And usually this negative positive mode switching is controlled by a simple electrical circuit. So if you can break that circuit and scan your mass spec independent of the ion source, then you can have access to almost 4 different modes, okay, of the mass spec. And again, okay, I'm thinking about a portable instrument that can now operate in 4 modes. Imagine that I have a mixture of 100 compounds. And these 100 compounds, okay, let's say I can detect 50 of them in the positive, 50 in the negative. But now take the 50 in the positive, then divide it again into 2. Now you have 25 here, 25 there, 25 here, 25 there. And so with a portable instrument with a small mass range, now I have a better chance of distributing the ions into more space. Now the resolution is not pushed anywhere. I just need a unit resolution to resolve a fewer number of compounds using the same portable instrument. So although the cost is the same, the value has increased. And so it's sort of in the reverse that we never think in that way. And again, this is possible because of the plasma. And we have a lot of references and calculations done showing that in fact the plasma can produce positive and negative ions almost in the same abundance. So all I have to do is to change the extracting voltage. Okay, do I want to take negative ions? I use positive extracting voltage to attract the negative ions. If I want positive ions, then I use negative extracting voltages to extract the positive ions. And I'm using the same mass spec, just a little trick now expands the approach. Okay, will this work? So when we started, we thought of a system that we were sure will work. So we took amino acid. It has an amino group and a carboxylic group. And so these two should produce positive ions for the amino and negative ion from the carboxylic. And so when you do positive, yeah, you see the N plus H. If you keep the source in the positive and you flip the mass back to negative, yeah, you see N minus H also. Likewise, if you do negative, the traditional negative is what you see. Keep the source in the negative, flip the mass back to positive, you see actually quite high abundance. And let me just say that this spectrum looks nice because we've background subtracted. Sometimes the background is quite high, like the next one I'll show you. Okay, so why is this important? I think I've kind of alluded to this already, but it's important because of these 5 points. Now, the first one is that I can get good molecular weight on a low resolution instrument. Why? Now, imagine that I have an unknown compound with 3, okay, in a positive is M plus H. The same compound is detected in the positive negative M minus H, okay? The same compound is detected twice. Traditionally, you can't detect the same compound in the positive or negative unless it's very unique of it. But if I can make it happen, and if I don't know M, I just do simultaneous equation, simple, and I solve it. So I don't need high resolution ICR to tell me that the thing is 0.000 thing. I can still do mass, my portable instrument, and get a reasonable information. I mentioned that the spectral complexity can be reduced, okay? Because if I have 100, I can split it into 25, 25, 25, 25, instead of 50, 50. That allows a better chance for low resolution instrument to do. And there are more of them here. In fact, we recently got a DOE funding to study plasma chemistry with this process. If the plasma really has two polarities of ions in there, and the mass spec can only detect one polarity when the plasma is in the cathode mode, then we are throwing away almost 50% of the information in the plasma. But now we can do, independent of the polarity in the plasma, we can scan the mass spec, and get data independently and get a full compositional studies done in plasmas, because we can detect the positive and negatives. Doesn't matter what source, is making the plasma. So obviously there's no time for all of this. I just want to demonstrate just one of them, okay, the molecular weight, to show you that we get more information even with a smaller instrument. And so you wouldn't expect that this fatty acid would do this chemistry, but it's actually doing it. Okay, my time is really going. Maybe I can end with, with this. I mean, with the first part of the studies. But here's a carboxylic acid, fatty acid. It turns out that if you do fatty acid, usually they tell you that for the acid, do in a negative mode. And in our source, if you do negative, this is what you see. So this is not background subtracted. It's messy, somehow a little messy. Okay, now keep the source in the negative mode, flip your mass spec to positive. Here's what you see. You see that all the noise, most of them are gone. And what you see here is a peak at 201, which you can relate very easily with the peak at 199. So if I didn't know the molecular weight is 200, these two peaks tell me right away that I have about 200 molecular weight. It works. Now we learned that, and if I were to go back and show you, we can pulse in vapor from this source. We learned that if you bring methanol vapor in the proximity of the source, you can actually change the carboxylic group into ester. And we've done a lot of work on this to prove that it's an ester group. And that ester shows up at 215. And so not only can I tell the molecular weight is 200, I'm also can tell that this is a carboxylic group, because we've done a lot of work, and amino acids don't react, with this ester. And so with my little mass spec, I can tell molecular weight, I can tell the functional group, and it turns out that even saturated fatty acids, okay, they are the ones that react with the methanol. The unsaturated ones with the double bonds, they don't react. So number 2 and number 3 don't give you the ester groups. Only number 1, 4, and 5 actually shows you the ester. And so in the same source, not only can I tell the molecular weight, not only can I tell the head group, I can also tell the backbone if it contains double bonds or not. And actually there's more. I can also tell you that the double bond is at number 6 or number 9 or number 10. And to do that, all you have to do now is to change your electrode. We usually use in silver, but if you change your electrode to iridium and you bring it into contact, okay, with the solution, the iridium actually acts as a catalyst. And if you apply a small volt, like 1.5 kilovolts, you form epoxide at the position of the double bond. And the epoxide shows up here. Now take this group and do MS on it. It fragments at the position, of the epoxide. And so these are two diagnostic groups. If you piece them together, it tells you the double bond was at number 9. Now this is oleic acid and number 9. If you take the isomer of oleic acid, which is vesenic acid, double bond is at number 11. Do the same analysis, do MS on the same peak. The peak you see are very different from oleic acid. You see two peaks that when you add them together, show you the position of the double bond is at number 11, but not number 9. So this is from the same source, doing molecular weight, doing hair group analysis, telling you that it's saturated or unsaturated. Now it even tells you the position, of the fatty acids as well. And so, well, that took longer than I expected, but at least the panoptic, which is an old English word saying all in one, that an all in one mass spectrometry, that allows you to increase the value of already existing instruments. Now, if I had time, maybe 10 or so minutes before 6, I can briefly show you how we are translating what we are learning here into the real world. And we are using malaria as an example. So maybe I should skip this, but malaria has been a big problem. There's been a lot of progress recently that even the WHO recommended that there were about 21 countries that could reach elimination by 2020. So all the blue regions were actually located, or they were identified as having promise to reach elimination. Well, we are in 2021, almost 2022, and only 2 of these countries, only 2 of them could reach elimination. Why? Because, yeah, they focus on the mosquitoes. Yeah, they spray, they go out of this to work, but then they took your eyes on the program a little bit, the thing goes up again. And these are very low density parasites that you need a highly sensitive tool to tell if somebody has only four parasites, or something. It's not a trivial issue. So now they have extended, this E2020 to E2030, and now they've opened up opportunities for people that have sensitive tools to come in and help. And so we recently got NIH funding to develop a method for malaria diagnosis in asymptomatic patients. And so again, I've already mentioned this. It failed, or it almost failed, because they were using this assured program. But now, maybe if we can use some equipment, we could get some results. And so my argument is that instead of focusing on just a simplicity, we should focus on the practicalities of the issue. Okay, we need sensitive tools to do it. Instead of focusing also on the cost, we should maybe look at the performance to cost ratio. I can reasonably give you a cost that actually gives you high performance. Collectively, I think that is a better approach than keeping the cost barely low, while you can't get any practical use from the device. And although mass spec costs just somewhat, our proposal is that if we can centralize a mass spec, millions of people can use the same device, and that actually brings the cost down. In fact, I have to argue that the device that works, this device that actually works in the field costs about $4 per test. Okay, when we go out to the field, we are testing about 1,000 people, okay, at once. That's about $40,000, for per sample. Okay, now if you do a replicate of it, 2-3 replicates, that is 40 times 3, almost more than what you can get for a portable instrument for a one-time use. But if you give me a portable instrument, I can use it over and over and over again and do replicates and get data, more sensitively. So all we are saying is, give me one mass spec, I can increase the value and bring down the overall cost. I'll set it, in a nearby town, okay, and I'll reach out to the villages, collect samples from here. And mind you, we are focusing on the asymptomatic people. They perceive themselves to not be at risk. They think they are okay, but they are the people that are carrying the parasite. In fact, 80% of the people in sub-Saharan Africa are asymptomatic. And the parasite likes that because they can keep on use them as a reservoir to cycle the disease. If we can identify these and treat them, then we have a chance of actually eliminating the disease. And so what we need for the portable instrument to work is we need an ion for the mass spec to detect it, but we need an inexpensive device that we can give to the people. And so we need a paper-based sort of thing. So for the ion, so we thought a lot about this. And we decided that the best way is to keep the platform the same. It's the same way you are dealing with the lateral flow assay. Put antibodies in the paper, put your blood on, capture the biomarker, now finish the immunoassay. But instead of using an enzyme for the immunoassay, we say we're going to use an ionic probe. This ion allows a mass spec to detect and the signal. The ion that we're going to detect, we synthesize it so we can control the mass. We can control the weight that will fit the portable instrument. And then we have a cleavable site that after we conjugate this onto the antibody and we freeze the immunoassay, all we do is we change the pH. This is very stable at neutral pH, actually surprisingly stable at low pH. But when you increase the pH over pH 10, this cleaves in seconds. We pick up this ion, then we detect it using our portable instrument. And so it's almost like a yes, no, but we are getting higher sensitivity because we are counting these ions much more accurately. Of course, we can put several of these ions in one antibody that also further increases the sensitivity. And so because we are not using enzymes, but it's just an ion, we are able to do the test in two different locations. We have two endpoints. We collect the sample at the remotest part of the regions. We don't need to analyze them because they are asymptomatic. They think they are okay. They have time to get the results later. We store these paper devices under ambient conditions, and then we bring it to a central lab. We use the mass spec for analysis. So the process, we can interrupt it. We can store it under ambient conditions. When we are ready, we restore the process and get the same results as if we did the analysis, in the same day. And the whole thing, I don't have time. It works in the microfluidic channel. We can split the blood into several different zones and get this going. So we've done this. And in fact, on the portable instrument, we are getting, about 200 picomolar. This sensitivity is actually reasonable for symptomatic patients. So we have a lot of work, some work to do. On the next slide, I will show you what we've done, but this is sensitive to about 200 parasites per microliter. For asymptomatic patients, we have to go lower. And so for lower, it's about 20 picomolar. And so just like 2 weeks ago, or maybe a month ago, and we are using this dendrimer so this 4G dendrimer has about 64 amine groups. That means at least we can put 64 ions, on this surface. And then we couple the dendrimer onto the antibody. So in the end, here's your antibody with a dendrimer with a lot of ions on it. So if you perform the immunoassay with this antibody, it brings a lot of ions with it. And thankfully, when we did this experiment, we are now down to about 4 picomolar, which is very comparable to what you get on a PCR. On the PCR, you all know that the amount of work you have to do to actually get the results. You have to control the temperature, you have to do extractions, and do all of that. In our case, it's a direct analysis. There's no sample prep. So now we are getting down to the 5 parasite molecular regime, which now permits us to go to the field and do analysis. We've used the device for clinical studies, and we can tell positive and negative samples. In terms of the stability, going from one place to the next, we've also tested it. And the antibodies on the paper before the assay is very stable. After the assay, they're also very stable. That means that the indigenous people, they don't need any special storage conditions. They can leave this paper, in their room, no refrigeration, do the test as they want. And when they finish the test, they can also leave it there for about 25 days, bring the test to us, then we will analyze and tell them. If you were to do this with enzyme, it actually, in the dry state, the enzyme degrades very quickly. Even in solution, about 7 days is all you've got. And at least we've got more than 25 days of stability. So at least without using enzyme, we get the stability and sensitivity to be able to reach people wherever they are, irrespective of their zip code, maybe even health insurance, because this is a cheap process that allows millions of people to be tested. Now we are working with this doctor. He's the one that introduced me to colorectal cancer. So we are expanding from malaria to colorectal cancer in underserved population within the US. I don't have time to go through the teaching staff, but just to say that I think we all know the flip classroom, when you give assignment to people, to students at home, and then they come to classroom to sort of ask questions about the assignment. That is not what I'm talking about. That is an inverted classroom. The reverse is a mirror image, okay, of the classroom. And so we are interested in this reverse, particularly because of the minorities. Okay, if we don't have people that look like them teaching them, if you don't have people that understands them from their perspective and be able to make honest connections, yeah, we are still not going to be able to recruit these minorities into the STEM. It's still a challenge, but I think the reverse, providing the mirror image to be an example for them is what we need, at least one of the things we need to make an improvement in this. And we've been working very hard in this. We've established teaching or research programs that we are reaching out to high school students. We've worked with a lot of minorities, students that are working in my lab that are interested. They are going on doing amazing work, because once they get exposed to this reverse idea, they become experts in themselves, and they go out and they preach to the choir, and they are winning awards here and there. So in all, I would say that thinking of research from the perspective of the recipient is something that we all could think about, and begin, to implement in our own small ways. I've been very fortunate to have funding that has allowed me to do these experiments, and the students also are very dedicated to their work. And I know I took a lot of time, but I'm happy to now answer questions and maybe dive a little bit more deeper into some of the areas that I glossed over. With that, I thank you, and I'll take any questions.
Great talk, Ibrahim. Nice. I got a couple of questions. Joe, do you want to go first? You got the chair. You should ask questions first, right?
Well, I think, I unfortunately, I have to, my children are going a little bit crazy upstairs, but I do want to give one, which I think is a good kind of starting off point for discussion, because that was an amazing talk. And all of that work was really incredible. And one of the things that kind of caught me is there are so many criteria for this panoptic mass spec and everything that you have to do to make it work. How do you prioritize that? And how do you tell a graduate student, okay, this is the priority number one, this is what you want to work on, and it's at the expense of everything else? It seems like there's just lots of things that have to fit together just right. it seems very challenging, but it's really amazing work here.
Right. So it depends on what you want to do. So I mean, all of these are together in a one box. So you decide. So instead of, at least in the traditional sense, you couldn't have HPLC, excuse me, APCI, working together with ESI. Of course, there are dual sources like that, but it's different in this way. So the graduate student, for example, will have to decide, as we all have to do a little research. That what do I have in my sample? Do I have a polar compound or non-polar compound? If you don't have that information, I always tell them, go back to whoever gave you the sample, and ask them, because it doesn't serve anybody good, to just work in the dark. And so once you have a little information about a sample, then you can decide, to do what you want to do. If it's a complex mixture, then apply some electrophoretic separation, pause positive, pause negative, and get results going. If you want to spread, this into multiple zones, just flip your mass spec, into plus or negative, and all of this is just a button that you are changing, right? It's not a complete switch of experimental setup. I think that's the consolation, I would say, that you touch a button, and you get results instead of bringing in with a different sample completely. Yeah. So that's what I would say, Joe, about this. Yeah.
Fantastic. Thanks very much. And if there are any other questions for Abraham, please feel free. I think Harsha is going to take it from here. Thank you.
Thanks, Abraham. Nice talk. Really liked it. I've got a few questions going back into your CRC, colorectal cancer diagnostics.
Oh, yeah.
You flashed a slide where you identified, or you're planning on detecting multiple analytes.
Yes.
Multiple antigens, I suppose. So what is the multiplexing strategy? Are you changing the length of the probe or detecting different masses all in one scan, one experiment? Or what is the strategy here for multiplexing? Are you, your probe has different length, or is it a different probe, or what is the chemistry behind that?
Yeah, that's a good question. So let me get back here. Get back to the Macrosampling Platform. So in this platform, I skipped it, so that might have. So you see, we have 4 test zones for each of these. What it is that, and when you put your blood, the same device will filter the red cells, and the plasma will get to here. And the black areas are all hydrophobic, and the white is hydrophilic. So when the blood or the plasma gets to the center, it wicks into the 4 different zones. So what you've done is you split the drop of blood into 4 zones. And you could store your reagents in here and get 4 different tests done, right? And so one way is, so this device was actually created for the malaria project, because we are going after 2 species of the malaria, the Plasmodium, falciparum, and Plasmodium vivax. But for the cancer, where we have 5 biomarkers, we can split this blood or serum into 5 test zones, where we will have different antibodies immobilized on each of the zones. Okay, now we could design the probe to have different masses. That would be sort of easy to do. The challenge is, in fact, when we started, it was very difficult to get the starting materials that had different length of this ionic probe. So we could get only 2 of them. And so we are stuck with that for now. But what we can do is that we can use the same mass, but detect it individually. So Okay, in this case, if you use the same mass, or I mean different mass, you could collimate the 4 test zones into one spot and analyze at the same time, right? And then the masses will tell you which antibody was active. But we could analyze these 4 individual test zones individually, although the masses are the same. That also will tell us, which spot was correct or not. And we are kind of leaning towards the individual analysis because of the limited starting material that we need for each test zone. Yes.
I see. But in the case of colorectal cancer, you have, like I saw in one of your slides, you had 4 or 5 different antigens.
Yeah, 5. Yeah.
5 antigens, right? So
Yeah.
it's conceivable that you can have a paper microfluidic device that can, and with 45 different probes.
Yes. Yes.
Do this analysis, right? Quite well.
Right. Yes. Right. So yeah, 5 different probes will be the easiest to do. In fact, we have another method which we are working on now. We haven't gotten any far, so I don't want to share it. But we can use different reactions that give different products. Okay, so this RNA probe only gives one product, but you can change the product if you change the mass.
Yeah.
But we can use a different reaction scheme. Let me see if I have it. I don't have it. Yeah, it's here. Yeah, we can do some photochemistry, and based on the starting material, we can get a different product. And so that also is something we are developing to enable multiplexing, while we are also kind of amplifying the signal. Yes, right.
Going back to your, you mentioned microdroplet chemistry where you're using headspace to derivatize sugars.
Right.
And then when you go back to your the source experiment, you said one of the analytes that you could not detect was a sugar.
Yes.
Is there some way you can couple using this method, this setup, to do kind of what Big Xer has done with droplet fusion, droplet chemistry?
Right. Yeah. So that was one of the things we actually want to do. So with this setup, yeah, we hope that we can fuse these 2 droplets together, and do have a chemical reactions happen while they are going to the mass spec. And one way that we've done it, we've actually done it, is to allow a vapor, to come in here. And this vapor could be like chloroform, which will ionize to give chlorides. And the chloride in the plasma, simply now attaches to the sugar. That also allows us to see it. And so I mean, all of that has been thought of, and we are just now working on them one by one.
I see. So what kind of reaction efficiency do you get when you do this droplet chemistry in adjacent sprayers like this?
For the fusion experiment?
Yeah, the fusion.
The fusion experiment. So for electrospray, and the fusion is very difficult actually, because electrospray uses high pressure of gases, right? So you notice that we are intentionally avoiding gases, because we want to go to the field and do analysis without carrying gas with us. But in the fusion experiment, in the traditional fusion, you are spraying your emitter at an angle, and one is pointing this way, other one is pointing that way. So you're only sampling a very small portion of where they fuse. It's not a very efficient process. We are hoping that with this, we haven't done much work on this platform yet, but with these droplets that are going, perpendicularly, but also spreading, hopefully we can get a better efficiency, but still, it will depend on the overlap, between the two droplets, and how much, you can actually overlap and get reactions going. Yeah, it's a challenge.
Mm-hmm.
Yeah, but the plasma might also help. So in fact, we have a paper coming up where we have plasma droplet chemistry. It's just a droplet that you fuse plasma into it. It doesn't work well for proteins, but it does work well for organic compounds. So maybe combining all of this together, in fact, I never thought about this until now. Combining the two droplets with the plasma could actually increase the efficiency,
Yeah. I mean, I have an interest in actually applying these methodologies towards biologics, right? If you can rapidly characterize, biologics, release glycan assays where you're actually detaching the glycan and looking at the glycans themselves, doing post-derivatization, real time. Those would be of real value even for folks in biopharma.
Right. Yeah. So in fact, I have to tell you that that first project I showed you here, that's where we are going. We've identified that the glycans are kind of pain, in general from our spec, and so this project we dedicated it purely for glycan analysis. It's surprising we could even tell isomers, of the glycans through some different reactions as well. Yes. Yeah.
Nice.
Yes. Yeah.
Thanks.
Yeah, you're very welcome.
I guess in the absence of Joe, I think Joe had to leave. Are there any other questions for the audience? I'd like to open it up. I guess we can go on forever, but in the interest of time, I guess,
yes.
Really interesting talk, Abraham. We really appreciate your time.
Oh, thank you. Thank you for the invitation.
New concepts and new ways of thinking. I guess that was as important as the science. So really thank you once again. And there's no other questions. We'll close it for today and hope to see you guys all in the new year. And thank you again.
Thank you, Asha.
All right. Bye-bye.
Okay, bye.